I'll be presenting on my paper titled Establishing a Breast Cancer Cell Line, Stably Expressing microRNA 4725 and Validating a Putative Target. This was done in WACCBIP and I was supervised by Dr. Lily Pemka. So breast cancer, as we know, as the #1 cause of cancer among women, and there is high mortality rate, especially in Africa. So we did microRNA, which is a post-translational gene regulators, were found to be implicated in breast cancer. And the rationale behind this was that they first did a study by Ho et al., saying about microRNA 4725, very specific in every tissue. So microRNA 4725, that was found to be upregulated in glioblastoma cell, that's cancer in the brain. So we wanted to do this study in breast cancer cells to know how it regulates some genes in the breast cancer cell lines also. So doing this study, we first generated microRNA 4725 stable cell lines by first culturing the cells, breast cell lines. I used MDMA 468. That was the breast cell line I used. So I cultured the cell for a period of 10 to 12 days in demand media. Then after, we seeded the cells so that the transfection can begin. So we did the transduction method with pyromycin, and we selected this antibiotic because we use a lentiviral that could inject the gene of interest, having the microRNA, and this lentiviral has antibiotic resistant gene. So it has pyromycin resistant gene. So we use pyromycin in selection to happen because if this breast cancer is able to stably get the cell lines in the microRNA 4725, it means when we use the pyromycin on them, they will not die because they're already resistant to it. And then in determining the expression of the putative target genes, there were 5 target genes that we looked at. So we transduced the cells using the lentiviral, and then we extracted RNA from it, and from the RNA samples, we did real-time PCR to know the expression of this microRNA and the effect that is having on the putative target genes. And then from the results session, this is for successful transduction and selection of clone. So I just made it multiplicate that 3 clones, but with the same treatment so that we can just pick one that was effective in these studies. So for clone 1, after pyromycin selection, you could see that the cell lines are still intact. And then for clone 2, after pyromycin selection, they all had the same condition, clone 1, clone 2, and clone 3. But this one was our control. These were the untransduced cells after pyromycin selection. So for that, we didn't inject the microRNA into them. So they didn't have the resistant gene. So after using the pyromycin on them, all the cells died. That showed that truly our transfection worked. This really showed our transfection worked. And this process that we are having is showing, figure 5 shows the microRNA 4725 expression in the cells. We wanted to know, before we can know the effect that this microRNA is having on the target genes, we want to know if first it was expressed in the breast cancer cell. So we just checked that, and truly it was expressed in the breast cancer cell. When we concluded on clone 3, we are using clone 3 for our studies because that was most where the transfection occurred. So getting to the expression of the microRNA on the putative target genes, first was aniline, and then KIF2C, NUSAP1, MKI67, and ASF1B. So all these putative target genes are found to be involved in cell proliferation. Most are found in cell proliferation. So we realized that after the expression and after the transduction process, the aniline gene, the 4 genes, aniline gene, KIF2C, NUSAP1, and MKI67 all had upregulation. Therefore, this microRNA we used, we found the effect being that it caused the upregulation of the genes as compared to the control. The control is the normal breast cancer cell and how this aniline gene is expressed in it. So after the transduction, we found out that because we injected the microRNA 4725, it made the aniline gene to be upregulated. It caused increased expression of the microRNA instead of this control where that's the limit that it should be. And it all happened in all the 4 genes that we checked. But for ASF1B, that is anti-silencing factor 1B, we found that after the transfection, all the genes were silenced as compared to the control. So realize that, you see, most microRNA, their function is they regulate genes and they silence genes. So realize that, yes, ASF1B is truly the target of this microRNA. But because it's been dysregulated, it caused upregulation of most of the genes that we found. So that was the observation we had about transduced cells maintain adherence and continue to proliferate even after pyrimycin selection. And then there was increased microRNA expression in the 4 genes that we found, except ASF1G, which has decreased expression. So in the future, I want to do whole transcriptome analysis of the clones to determine molecular pathways involved in microRNA gene expression. This will help us get the full picture of the microRNA transcript and see how it's able to regulate these genes that we are targeting. So these are some selected references, and it was funded by WACCBIP. Thank you so much.